Hello, my name is Kristopher Hite, and I'm a postdoctoral research fellow in the biology department at Emory University. This fall, I'll be teaching genetics in conjunction with Dr. Valerie Odero-Mara at Clark Atlanta University. Today, I want to talk about the process of cellular reproduction, namely the cell cycle, or the timeline with which a cell undergoes reproduction, and mitosis, which is the process by which the genetic information inside a cell is separated. So in order for cellular reproduction to be successful, three things need to happen. The DNA, the genetic information inside the cell, needs to be copied. Those copies need to be separated, and the cell needs to divide. What we're seeing here is the embryo of a worm, a C. elegans, undergoing initial stages of development. And we see that the initial stages of development from a zygote, a single cell, into an adult organism are just a series of copying events or cloning events. Cloning sometimes used as a scary word in the media, but really it just means to make a copy. And so all multicellular organisms make millions, if not billions and trillions of copies of their cells throughout their lives. The reason that cell reproduction is so important to understand is that it's essential for growth of all multicellular organisms. As we see here, this Australian tree fern is using cellular reproduction to unfurl its fronds. The same is true of the developing dog embryos to take a puppy into an adult dog, or you and me, a human being going from a baby to an adult. It all happens because of cellular reproduction, the copying or cloning of cells. So let's jump right into the cell cycle. So what is the cell cycle? This circle that's often used in textbooks is basically a timeline of the cell's life. We start here at the G1 phase, which G1 stands for gap 1, but I remember it by thinking of it as growth, because at this stage, the cells only have one copy of all their chromosomes, and they are just growing. They're making all the cellular machinery, the proteins, the RNA, everything that they need to survive and thrive is happening here in G1 phase. Now, late in G1 phase, they hit this G1 to S transition. Now, once they hit this transition, they're dedicated to making copies of themselves because they've started to make copies of their genetic information, their DNA. So this S phase, the S here stands for synthesis. During S phase, the DNA in the cell is copied. Now, after S phase, they go into this G2 phase, which is where they ready themselves for mitosis or cell division. So the timeline for a average human cell is 10 hours in G1, about 9 hours to complete the synthesis of DNA, and then about 4 hours in G2. Now, mitosis as a process can happen pretty rapidly, just half an hour to an hour. And in the next part of the discussion, I'll just break down the stages of mitosis. Forgot to mention here, some cells can actually jump off the cell cycle here and enter this G0 phase. Cells like your nerve cells, your neurons, and your muscle cells go into this G0 phase at a certain point of development in which they basically don't actively divide anymore. So mitosis happens in a series of steps. Now, everything that I just described just now, G1, S, and G2, those are all components of this first phase, the interphase, which is the longest phase usually of the cell cycle. And then the rest of the cell cycle, or mitosis, happens in a series of steps: prophase, prometaphase, metaphase, anaphase, and telophase. Now, I usually use a mnemonic to remember these things. I know it's tough to memorize things, but this is something that you're going to need to know. So if you can just remember IPMAT, that stands for interphase, prophase, metaphase, anaphase, telophase. Now, the one caveat there is you got to remember that some textbooks list it, some textbooks don't. This prometaphase, which is the stage right before the chromosomes align on the metaphase plate, just means pro, meaning before metaphase. So you got prophase, before metaphase, prometaphase, metaphase, anaphase, where the chromosomes begin to separate. That is when the sister chromatids separate. And then telophase, followed by cytokinesis. Now, cytokinesis is kind of its own separate event whereby the cells actually pinch off from each other. And that's a really neat process that I'll show you a video of. So I want to thank Jennifer DeLuca at Colorado State University for responding to an email that I sent yesterday requesting a video that I remember seeing of these rat kangaroo cells that she was using to study these kinetochore complex proteins. So here goes the one of these rat kangaroo cells dividing. See all the chromosomes lining on the metaphase plate and dividing. Let's watch that one more time. Oop, there you see cytokinesis and separation of the cells. Let's watch that again. Oh, it's so cool. So cool. All right, thanks a lot. Now, I'll be available on the internet to answer more questions. You can refer to your textbook to learn more about the specific terminology. I really get excited talking about the different checkpoints throughout the cell cycle, especially the spindle assembly checkpoint. There's a lot of amazing molecular mechanisms that act as like a surveillance system to make sure that all the chromosomes are aligned on that metaphase plate before anaphase begins. So yeah, I invite you to come and join the Facebook forum and discuss these things further so we can work out the nitty-gritty. Thanks for joining me. Kristopher Hite, signing off.